Hello, and welcome to our section about lipids. You've probably heard that word before. If you remember, our phospholipid bilayer has lipids in it. Lipids are basically just referring to fats and oils. So if you remember all of our organic compounds in living cells, we just talked about carbohydrates in the last section, and today we're talking about lipids. Two more to go after this. We'll talk about proteins and nucleic acids. What are the function of lipids? Well, lipids are important for long-term energy storage. Lipids are also the structural building blocks for cell membranes. Remember our phospholipid bilayer? Those are lipids that make up that membrane. Here are just some examples of some lipids. What is the structure of a lipid? Well, we call the simple fats that we find in our foods and in our bodies triglycerides. Sounds like a really big word, but it's not too difficult. We have the TRI, tri on that word, meaning three. Glyceride is going to refer to the fact that the three fatty acids are held together by a glycerol molecule. So triglyceride means we have three fatty acid chains, and then we have a glycerol that binds those three to itself. And if we look at what that looks like chemically, we can explore that down here. The glycerol molecule looks like this, and the fatty acid chains look like this generally. And when we have our, remember, each of those corners represents a carbon. Okay, we just didn't write them all out, but those are all carbons in there. And we also didn't write out the hydrogens that are bonded. There would be two hydrogens bonded onto each of those carbons as well, but it's not written out. This is just a simple form so that we don't have to write all of those carbons out in longhand here. But this chain is our fatty acid. When we have three fatty acids, here's 1, 2, and 3, they can bond to this triglyceride molecule, or to this glycerol molecule, and we get a triglyceride. That's just another way of writing this. I expressed this out, showing you the actual chemistry involved and the different atoms present. So we have some different types of fats. I'm sure you've all heard of saturated and unsaturated fats. Well, what's the difference? First, let's look at saturated fats. You probably know the saturated fats are the bad ones, and you're not supposed to eat a huge number of them. They tend to be solid at room temperature. Examples of saturated fat would be butter and lard. Unsaturated fats tend to be oily liquids at room temperature, and here are some examples: olive oil, corn oil, and sunflower oil. Now I'd like to talk about it a little bit more from a chemistry standpoint. Unsaturated fats are called unsaturated because they contain at least one double bond within the fatty acid chain. So let's look at our triglyceride right here. We have the 1, 2, 3 fatty acid chains. Wow, what's happening here? This one's bent. It's because there is a double bond between those particular two carbons, and it's called unsaturated because when the carbons have a double bond in between them, there is 2 fewer hydrogens that are bonded. Carbon can only form 4 bonds at one time. And so 1, 2, and then because this is a double bond, that's 3 and 4 bonds. Can't bond to anything else, so we lose some hydrogens here in the process. So when we talk about unsaturated fats, we are talking about, it's called unsaturated because it's unsaturated with hydrogens. We're taking some hydrogens away. And in replacing those hydrogens would be this double bond between 2 of the carbons. And it does a few very interesting things in our molecule. It bends it. It makes it kind of crooked, if you notice. So that is the reason why oily liquids, the unsaturated fats tend to be oily at room temperature, because this long chain is no longer straight and it can't pack and condense tightly with other chains of fatty acids. It's bent, and especially when I start getting multiple bends in here, these molecules no longer stack tightly and compactly and neatly. So there's more space in between them, and they tend to be liquid rather than solid at room temperature. Now, I'm sure you've heard of monounsaturated fats and polyunsaturated fats. Those basically mean, for monounsaturated, that our fat contains only one double bond. So right here is a monounsaturated fat because it has one double bond. A polyunsaturated fat contains more than one double bond. So it would have more than just this one here. There would be several in that fat. Types of fats. We have more types of fats here. We have phospholipids. You know about, because our phospholipid bilayer, this should be review for you. If you recall, in the cell membrane, we have our phospholipid bilayer where we have the hydrophobic tails and the hydrophilic heads, and that forms the cell membrane. When we look at that structure a little bit more closely, we will see that those tails are actually two fatty acids. Those are the tails. Here is our glycerol molecule, like we're familiar with now, and a phospholipid includes a phosphate group right here. It happens to be a chemical component that has phosphorus and oxygen in it. So the phospholipid structure has two fatty acids. As for tails, it has one glycerol molecule, and it also has a phosphate group. Remember our phospholipid function. This is all review as well. The nonpolar tails form the interior of the cell membrane. The polar head associates with the water, and it forms a bilayer, which acts as a selectively permeable cell membrane in conjunction with proteins as well. And it regulates which molecules enter and exit the cell. Another type of fat that's very important is cholesterol. I know you've heard this word before. What is cholesterol? Here's a picture chemically of what cholesterol looks like. A little bit different than what we've been looking at for our fats, isn't it? Cholesterol happens to have 1, 2, 3, 4 fused rings involved. So it's a little bit different as far as its chemical structure. It has 4 rings, and then it has the side group here. And cholesterol, if you look at this, it is redrawn in this picture, right here, and here, and here, and here, and throughout. That is just a redrawing of this molecule. Again, carbons are not shown. Right there those would be all carbons, and they're not shown in this way that we draw the molecule. But if we look at this diagram, this is a phospholipid bilayer. We have our heads and our tails, and what is in between some of these phospholipids? Cholesterol. It's a different fat, and it's part of the lipid bilayer. And its function is to maintain proper permeability and fluidity. It helps make sure that the lipid bilayer can move easily and that things, different materials enter and exit the cell as they're supposed to. It is the component in animal cell membranes. It's also, in addition to being an important part of the animal cell membranes, it is a precursor for synthesis of hormones and other vital substances. What is that? What am I saying you? Precursor. If you've never heard that word, it just means it is the starting material for something. Synthesis means to make. So cholesterol is the starting material for making hormones, including sex hormones. It also makes other vital substances in our bodies. So cholesterol is actually a very important fat that we need in our cells. Advanced, You could look at what are some of the specific substances synthesized from cholesterol in animal cells. Maybe look at the chemical composition of some of the hormones. What is a hormone? What does that look like? What other questions do you have about lipids and living organisms? Research your answer. Come and tell me, and I look forward to seeing what you find out. We'll see you in class.